Protein production. As it has been mentioned before, depending on the characteristics of the protein and available resources, the expression system chosen to produce the recombinant protein is different. As examples, here we're going to show some clues for protein expression in E. coli and mammalian cells. Protein production in E. coli. As an example, we are going to show the steps to produce a specific protein in 2 liters of E. coli batch culture. Since this is a high volume, first is necessary to prepare a pre-inoculum. In one flask, 100 ml of the culture medium, in this case Luria-Bertrani LV, are added. The antibiotic, in this case ampicillin, is added to the medium to selectively allow the growth of cells that have the plasmid with the antibiotic resistance. The bacteria containing the plasmid with the transgene of the protein to be produced are stored at -80 Celsius degrees. The frozen bacteria are attached to these small beads, and is enough to add one of them to the culture medium. The flask is incubated overnight under agitation at 37 Celsius degrees. The next day, an evident bacterial growth can be seen in the pre-inoculum. The level of growth is determined by the optical density of the sample at 550 nanometers. The 2 liters of culture medium are prepared adding the corresponding antibiotic. Later, the inoculum is added to have a final optical density of 0.1. Then the flasks are incubated at 37 Celsius degrees, and the optical density is checked several times until it reaches a value between 0.5 and 0.7. At this optical density, we assure that bacteria are at the beginning of the exponential phase of the growing curve. This is the best moment to induce the protein expression. For non-constitutive expression systems, an inducer must be added to the medium to induce the protein expression. In this case, because the transgene is under the control of the LAC operator, we use IPTG as an inducer at 1 millimolar final concentration. Then, the flasks are incubated at the appropriate temperature and time to favor the best protein expression, normally desired in high amounts in a soluble form. In this case, the cells are incubated overnight at 16 Celsius degrees. The next day, the optical density of the culture is measured in order to check the correct growth of the cells. The culture is centrifuged 15 minutes at 6,000 Gs to pellet the cells. The supernatant in this case is discarded since the recombinant protein produced is contained inside the cells. The pellet is then resuspended in PBS to be washed and centrifuged again in small tubes. The obtained pellets are frozen at -80 Celsius degrees until the purification day. Protein production in mammalian cells. Here we are going to show the most usually transfection method used in our service for transient protein expression in mammalian cells. 4 different components are necessary to carry out the transfection procedure: the cells, the culture medium, the DNA, and the transfection agent. For protein production in mammalian cells, we normally use HEC293F cells, coming from the human embryonic kidney cells that have been adapted to grow in suspension in a particular culture medium. These cells are adapted to grow at high cell densities and have a very transfection efficacy in large volumes. The serum-free expression medium freestyle 293 is used for cell growth, and at the same time is switchable to perform the transfection, avoiding in this way to change the medium. The DNA to be transfected is an expression plasmid for mammalian cells with the gene encoding the recombinant protein to be produced. A secretion peptide signal is added in the N-terminus for proteins that are needed to be secreted to the extracellular medium. The vector contains a promoter that drives the expression of transgenes, a translational signal, the GOSAT sequence, a prokaryotic replication origin, and a selection marker for the selection and propagation in prokaryotic hosts to replicate and isolate the plasmid in high amounts needed for the transfection. Polyethylenamine, PEI, is an organic cationic polymer that functions as a transfection agent vehicle to introduce DNA inside mammalian cells. PEI interacts with DNA through electrostatics interactions, forming polyplex nanostructures that interact with the plasma membrane and are introduced inside the cell through endocytosis. PEI is the most common vehicle used for gene delivery since it has a high transfection efficiency in many mammalian cell types with minimal cytotoxic effects. To start with the transfection procedure, first it's necessary to look at the cells that are going to be transfected. An aliquot of the culture is taken and mixed with the vital dye trypan blue that stains dead cells. Cells mixed with the dye are introduced into an Neubauer chamber to determine their concentration and viability, and stained cells with a birefringent round shape will indicate that cells are in good conditions. The transfection normally requires a cell concentration between 0.8 and 1.2 million of cells per milliliter. Once checked that the culture has the appropriate concentration of apparently undamaged cells, the transfection procedure can be started. A transfection mixture must be prepared in a volume representing 1/10 of the total final culture batch. This means that, for example, for this particular case, half a liter of a final culture is composed by 450 milliliters of cultural cells plus 50 milliliters of the transfection mixture. In a tube with 50 milliliters of culture medium, the plasmid is added at a ratio between 0.51 micrograms of DNA per milliliter of cells. Later, the transfection agent is added in a proportion of 3 milligrams of PEI per milligram of DNA. The mixture is vigorously mixed and then kept resting during 10 minutes at room temperature to allow DNA PEI complexes to form. After the 10 minutes, the mixture is added slowly to the cell culture and under agitation in order to distribute it homogeneously, avoiding in this way high local PEI concentrations that are toxic for the cells. The culture is then incubated under agitation at 37 Celsius degrees and 8% carbon dioxide. Between 4 and 24 hours later, a final concentration of 4 millimolar valproic acid is added to the culture to enhance the transgene expression by promoting histone hyperacetylation. The number of days in which the culture is kept under incubation depends on the information obtained in previously performed time course expression tests. In these tests, the level of protein expression is determined day by day to look for the moment that gives the best amount of protein in the desired compartment. Once the incubation period is over, the culture is centrifuged 15 minutes at 5000 Gs. For secreted proteins, the supernatant is kept, and the purification is carried out immediately from the extracellular medium. In case of proteins contained inside the cells, the pellet is frozen at -80 Celsius degrees until the purification. Now we move to the next section, protein purification.